ब्रह्मनाथ ओ शांति 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 स्वस्थ। Hello students, in the previous class we discussed about the classification of animals part 1 in which in detail we discussed various levels of the organization. First we discussed the cellular level of organization, next we discussed the tissue level of organization, further we discussed the organ and the organ system level of organization. Next we discussed the body plan, under which we discussed the cell aggregated plan, then the blind sac plan in which there is only one opening which helps in both the ingestion and the egestion. Further we discussed tube within tube plan in which two openings are present, one is your mouth, another one is your anus. Mouth helps for the ingestion of the food and the anus helps for the egestion of the undigested food particles. Further we discussed that under the body, under the tube within tube plan, there are two types of organisms. One is your, we can say, the protostomic, other one is your deuterostomic. The organism during their embryonic development, in which the digestive tract, the digestive tract of formation, first the mouth is being formed. Okay, then that is known as your, we can say, the protostome or the protostomic animals or organisms. Or we can say the blastopore, which is first developed into the mouth, then this is the case of the protostomic, and in which the blastopore develops into the anus first, or in, we can say, in the digestive tract first the anus is being formed. It is the case of your deuterostomic. Further we discussed the symmetry, in which we discussed two types of symmetry. One is your asymmetrical, other one is your symmetrical. In the symmetrical we discuss further two types of symmetrical. One is, or we can say, the radial symmetrical, and also then we discuss the bilateral symmetrical. So this much we discussed in our previous class. Today we will discuss our next segment of the classification of animals, or we can say classification of animals part 2. The first, the next, the fourth one, first, second, third we discussed. Today the fourth one, germlets. In the multicellular animals, during the embryonic development, the cells are generally arranged to form the germlets. Means what? When the fusion of the male and the female gamete happens, as of now, you just listen. Don't worry about of the concept or something that this is the clear concept is in your second year. Just listen what I am saying. When the male gamete and the female gamete fuse, it forms the zygote. Simply the zygote is a single cell stage. Further it divides to form 2 cell stage, 4 cell stage, 18, 16, 32, etc. it being forms. It forms morula, zygote from the morula, blastula, lastly gastrula it being forms. In the gastrula, the rearrangement of the cells takes place, by which 3 germlets are being formed: outer ectoderm, middle mesoderm, inner endoderm. Okay, from these various germlets only, the ectoderm, endoderm or mesoderm, various parts or organs are being formed, by which the complete organism is being formed finally. Okay, what is germ layer? So we can write in the germ layer, in the multicellular animals, we can write the cells are being arranged to form during the embryonic development to form the germ layers. Okay. So we will write in multicellular organisms, we can say during the embryonic development, during the embryonic development, cells are arranged to form the 3 germ layers. Okay, whereas your outer one is your ectoderm. Outer one is your ectoderm. Okay, middle one is your mesoderm. And the inner one is your, we can say, endoderm. Endoderm. Okay, remember, inner one is your endoderm, middle one is your mesoderm, and the outer one is your ectoderm. So, what is diploblastic? In the diploblastic animals, 2 germ layers are present. Outer ectoderm is present, inner endoderm is present, and in between the ectoderm and endoderm, an undifferentiated layer is present, that is called a mesoglia. Means undifferentiated means what? It cannot be, we can say it is an ectoderm, or we cannot say it is an endoderm, or we cannot say it is an mesoderm. It is an undifferentiated layer. So it is the undifferentiated layer, that is your mesoglia, which I have done in the red color. It is being present in the between the ectoderm, outer ectoderm, and the inner endoderm. Here we can see the outer one is your ectoderm, inner green color is your endoderm, and middle undifferentiated layer is your mesoglia. Okay, so we can write in diploblastic animals, 2 germ layers are present. Okay, outer ectoderm and inner endoderm. Simple. Apart that, an undifferentiated layer is present, that is called a mesoglia. An undifferentiated layer is present in between the ectoderm and endoderm, that is your, we can say, mesoglia. Understood? Now the triploblastic, similarly, example, in the here example we can say porifera, okay, coelentrates. Okay, next here, triploblastic, simple. In the triploblastic organism or animals, they have 3 germ layers. Outer ectoderm, okay, middle mesoderm, inner one is called endoderm. Simple. And in this, we can say the example, we can take the first triploblastic organism, we can say the Platyhelminthes, or we can simply take example Platyhelminthes to chordata. So we can say here in triploblastic animals, 3 germ layers are present. Okay, outer ectoderm, middle mesoderm, and inner endoderm. Okay, and here we can take example Platyhelminthes up to chordata. Here we can take example Platyhelminthes to chordata. Okay, all the phylum we'll read in detail, don't worry about that. So germ layer we discussed in the multicellular organism, the development during the embryonic development, the cells are arranged to form the germ layer. It is of three types: ectoderm, endoderm, mesoderm. In the organism in which two germ layers are present, outer ectoderm, inner endoderm, then those are called your diploblastic. Examples are porifera and cylindrites. Whereas in the organisms in which three germ layers are present, those are called triploblastic. Okay, outer ectoderm, middle mesoderm, inner endoderm. Common example we can take from the platyhelminthes to the chordata. And here we have the diagram. In the diploblastic, have the outer ectoderm. In middle, the undifferentiated mesoglia present between the ectoderm and endoderm, and the inner endoderm. Whereas the triploblastic, outer ectoderm, middle mesoderm, and inner endoderm germ layer. Okay, please note down. Students, next we will discuss our next classification of animals, which is very important, that is your Cillum. The Cillum means what? Simply we can say it is a body cavity. Okay, it is a body cavity which is present in between the gut wall and the body wall, which is further lined by the mesoderm. Okay, first I am writing, then I will make you understand. Cillum is the body cavity present between the, we can say, the gut wall and the body wall, which is lined by mesoderm or mesodermal layer. So, students, what is Cillum? Cillum means we can simply say a cavity or space within the body. See, suppose this is the, we can say this is the outer M structure of the organism. Suppose this is, we can say, the alimentary canal. Okay, starting from the mouth, ends with the anus. So, simple, if it is the alimentary canal, so it is also known as your gut. Okay, what is said? It is the gut. So the wall of the gut is simply said as the gut wall. Okay, and this is the outer layer, or we can say outer covering, that is known as your body wall. So the space between the gut and the body wall, that space is nothing but your Cillum. Okay, as you know, this is the tube within tube plan. Okay, we discussed tube within tube plan. Suppose this is a tube within a tube. Okay, the body wall is another tube, and this one is a tube, the alimentary canal or the gut. So tube within tube plan. Okay. So the space between the cavity between the gut wall and the body wall is nothing but your Cillum. But it is lined by the mesoderm. The criteria is it should be lined completely or layered completely by the mesoderm. Okay. So what is Cillum? It is the cavity present in between the gut wall and the body wall, and it should be lined by the mesoderm layer or mesoderm. Next, what is, we can say, the coelomate? Come to here. Coelomate. This one is the case of pseudocylomid. This one is the case of acoelomid. See, coelomate means the animals which have the true coelom is nothing but your, we can say, the coelomate. See, this is the case of the coelomate. Okay, the animals which have true coelom. What do you mean by true coelom? Means that coelom should be properly lined by the mesoderm. See, here the outer is one is your ectoderm. Okay, middle one is your mesoderm. Okay, this green one is your endoderm. Okay, and this one is your gut, or we can say primitive gut. As we are studying in the embryonic stages, we can also write is your primitive gut. Means it is the stage of before forming the gut. So you can see, students, I have given the black color to the ectoderm, red color to the mesoderm, and the green color to the endoderm, and the empty space is your gut. This space, students, you are seeing, okay, this the gut we are taking in the transverse section. Okay, this section we are showing as a dislike. Okay, so this is the cavity. Our first criteria is completed. As I said, true cillum means it should be lined by mesoderm. You can see, students, externally and internally both it is being lined by the red color mesoderm. So it is the coelomate, and the gut is present. Gut is present, cillum is present, and the cillum is entirely lined by the mesoderm. So we can write the animals which have true cillum is called as coelomate. Simple example we can take the annelids, echinoderms, chordates, annelids, echinoderms, chordates. Okay, simple. Pseudocylomid means, you know, students, pseudo means false. Cillum is present, but it is not the proper false. So we can proper cillum. So we can say the pseudocylum. See, what I have told you, students, that the cillum should be properly lined by the mesoderm. See, here ectoderm is present. Simple. Endoderm is present. Simple. Okay. This one is your pseudocylum. Why pseudocylum? I will tell you. This one is your mesodermal pouch. Mesodermal pouch. Okay, mesodermal pouch. So this one is your cavity, this space is your cavity, but what I have said, the cavity must be lined by the mesoderm. Does it is lined by the mesoderm? No. Instead, some mesodermal pouches are present, not properly developed mesoderm is present. Some undeveloped, or we can say mesodermal pouch is present inside the, we can say, the cavity or the cillum. That is why it is known as a pseudocylum. And the animals which have the false cillum, or we can say the pseudocylum, are known as a pseudocylomate. Okay. The animals which have, or we can say which don't have, it will be more appropriate, which don't have true cillum are called as, you can write are called as pseudocylomate. Pseudo means false. Cillum is present, but it is not properly lined by the mesoderm. Pseudocylomate. Okay, examples: Nemathelminthes. Okay, you can see the complete diagram. Next, gut is present, and this one is your gut. Okay, this one is your gut, or we can say primitive gut. Okay, remind primitive gut. Next, acoelomate. Students, when you apply a term A means it is completely absent. Means acoelomate means cillum is completely absent. So the animals which don't have any cillum, those are called the acoelomate. The animals which don't have cillum. Students, don't get confused. I am here written the animals which don't have true cillum. Here I am writing the animals which don't have cillum. Okay, so these are two different things. I am writing if this does not contain true cillum means it containing false cillum, means the coelom in which the mesodermal lining is not present. That is why I written the true coelom is not there. But here I am writing the coelom is completely absent, or we can say the animals which don't have coelom are called acoelomate. Example, Platyhelminthes. Okay, students, see here gut is present, simple. Endoderm is present, mesoderm is present, ectoderm is present. But can you see any type of sort of space? Can you see any sort of space in between the gut and the body wall? No, students. So here no coelom is present. That is why it is called the coelomate. The acoelomate. So simple, students. The coelom, as we discussed, it is the body cavity in between the gut wall and the body wall. Okay. It is of three types. If true coelom is present, coelomate. If true coelom is not present, then pseudocoylomate. If coelom is not present or absent, then it is called acoelomate. Okay. Examples are Enterosilix, Equidomes, Chordates in case of coelomate, Nemathelminthes in case of pseudocoylomate, Platyhelminthes in case of the acoelomate. Okay. And these are your diagrams. Please note down. Students, one labeling you do here, this is the coelom. Okay. As we have done here, this one is pseudocoylom, this one is coelom. Okay, just do one labeling. This one is just coelom, right? Students, next we will see how the coelom are being developed. Accordingly, in the various two ways, the coelom are being developed. In two ways the coelom are being developed. One is your, we can say, the Biswajyoti coelom, other one is your Enterosilix coelom. Means these are the type of coelom on the basis of how they are being developed. Biswajyoti means simply we can say splitting of the mesodermal cells. Next is Enterosilix elements. The archenteron makes pouch-like segments and then they split. See, students, how it is formed. As we discussed up to the blastopore stage, as I told you, means the zygote, the morula, blastula, then the cells are rearranged to form the three germlets. Outer black one, that is your ectoderm, middle red one, that is your mesoderm, and the green endoderm. We discussed this in detail, so I am not doing any sort of labeling now. Okay, you can understand. This is also the pole-like segment of blastopore, and this one the cavity of the gastrula is your archenteron. So students, this happens in the embryonic stages. When if the type of coelom is being formed by Biswajyoti, means we can say splitting. You can see these are the mesodermal cells. If these mesodermal cells get split, these mesodermal cells that get splits, now this type of, we can say, coeloms are being developed. Okay, or a cavity is being formed. This one is your gut. Okay, this one is your gut, and the mesoderm are being split to form the coeloms. Okay, next it is now further developed. As I said, a true coelom should be lined by the mesodermal cell. Okay, so it is being lined by the mesodermal cells, and these are the developing coelom. Finally, it will be completely developed into such like structure, having the outer ectoderm, middle mesoderm, inner endoderm, and this center one is called the primitive gut. Listen, students, I am written only gut, but it is basically primitive gut. Okay, means the primitive stage of the alimentary canal. And this cavity, as you can see, is the true coelom or the coelom. So the type of coelom in which the coelom is being formed by the splitting of the mesodermal cells, this is called a Biswajyoti coelom. Okay, then the enterosilic means we can say the alimentary canal or the primitive gut or the gut. See, as this one also you can know, ectoderm, mesoderm, and the green one is the endoderm. If what happens here, blastopore is also present. What happens here, the archenteron in this case forms a bulging like structure all called the pouch like segment. You can see here some pouch like segment 2 is formed, and that pouch like segment will further get separated, okay, or split. This pouch like segment get separated to start forming the cilium, and this will further develop as the developing cilium which is lined by the mesoderm, and finally it will be completely developed into a true cilium as having the cilium, outer ectoderm, inner mesoderm, middle mesoderm and inner endoderm. So as we know, the cilium is the cavity which is present in between the body wall and the gut wall. Okay, so that is known as your cilium, but it should be lined by the mesoderm. As you can see, it is properly lined by the red color mesoderm. Here I have taken the black color is your ecto, green color endo, and the red color is your mesoderm. So students, clearly understood, archenteron means it is the embryonic gut, means it is a primitive gut. Okay, in the gastrula stage the cavity is called the archenteron, or it is also known as the embryonic gut, which further leads to formation of the alimentary canal or the digestive tract. Next, so students, so the type of cilium in which the mesodermal cells are split to form the cilium, these are called the schizocilic cilium. Enterosilix cilium occurs in our case or human case. The pouch like segments are formed in the archenteron. Further, the pouch like segments are being split and it is being developed to further form the true cilium. Okay, so this is the cilium which is formed from the basically the archenterons are formed by the pouch like segment, and then by its split to form the cilium, then it is known as the enterosilic cilium, which occurs in our case. Okay, so this is the type of cilium in how the cilium are being formed. There are two types, schizocilic, enterosilic cilium. Okay, please note down. Students, now come to your next two classification of the animals. First one is your segmentation, then the sixth one is your segmentation, the seventh one is your notochord. Segmentation. In some animals, their body is, we can say, is divided externally and internally into various segments containing various, or we can say the same repeating organs. So we can write, in some animals the body is divided externally and internally into and internally into various segments having the same repeating organs. Okay, having the same repeating organs. So means what? Suppose we are taking the case of the earthworm. See students, it have various, the body is not only externally divided but is also internally divided. So that is why in the same organs are also present, repeating organs are also present in the case of the segment. Suppose this one is a segment, means that organs that are present in this segment are also present in this segment. So the body in some organisms or some animals, the body is externally and internally divided into various segments which contains some repeating organs. So we can write, in earthworm this pattern is of segmentation is present. This pattern of segmentation is present, called the metameric segmentation. Obviously, the phenomenon is known as a metamerism. The phenomenon The phenomenon is known as metamerism. Okay, and the individual segment are known as your metamer. If you are taking the plurals, then metameres. The individual segments are known as metamer. Okay, students. Here we taken in the case of the earthworm. Next, notochord. What is notochord, students? It is simply a rod-like structure developed from the mesoderm, which forms the dorsal side of the animal, or we can say dorsal side of the body. Remember, students, this one is your ventral sides always, this one is your dorsal side. So the notochord, which is a rod-like structure which developed from the mesoderms and forms the dorsal side of the body, is known as your notochords. In which the notochord is present, they are the non-chordates. Okay, starting from the porifera to platyhelminthes, and in which the notochord is absent, those are the present, that are called your chordates. So notochord is a rod-like structure developed from the mesoderm and forms the dorsal side of the body. Okay. Example we can write from the porifera to echinoderms, we can write, not platyhelminthes, up to echinoderms. Dorsal side of the body. We can write there are two types, non-chordates and chordates. Students, in the non-chordates means notochord is absent. Notochord is absent. Chordates means, student, notochord is present. Notochord is present. In the non-chordates, we can take the phylums from porifera to the echinoderms. To the echinoderms. Simple. So the notochord means it is a rod-like structure which is developed from the mesoderm and forms the dorsal side of the body, students. If the notochord is absent, then it is called the non-chordate. If the notochord is present, then it is known as the chordates. In the non-chordates involves the phylums starting from the porifera up to the echinoderms. And the segmentation we discussed. The segmentation means in some animals or some organisms or animals, the body is externally and internally being divided into various segments which contains the same repeating organs. Okay, and suppose in the we are taking the earthworm case, this pattern is present. This is called a metameric segmentation. Okay, and this phenomenon is called a metamerism, and the each individual which or the segment are known as your, we can say, metamer, or the plural is your metameres. Okay, students. So today we discussed important classifications. Okay, so first we discussed for the various classification we have discussed. Okay, so we discussed the Cillum, we discussed. Okay, and also important segmentation, notochord, then the germ layer also we have discussed. In the germ layer means in the embryonic stage, the cells are being arranged to form various germ layers: outer ectoderm, middle mesoderm, and the inner endoderm. Okay, if two layers are present, means the ectoderm and the endoderm, then it is known as your diploblastic. If three germ layers are present, ectoderm, endoderm, mesoderm, then it is known as your triploblastic. Okay, students. Next we discuss the cilium. It is the cavity in between the gut wall and the body wall. It is of, we can say, we discussed it is of various types, such as your the coelomate, the pseudocylomid, or the acylomate. In the coelomate case, true Cillum is present. Pseudocylomate case, the true Cillum is absent, and in the case of acylomate, complete Cillum is absent. Okay, true Cillum means if the Cillum is completely lined by the mesoderm, then it is called true Cillum. False Cillum means various mesodermal pouch are present and it is not lined by the mesoderm. Okay, simple. And next we discuss how the Cillum are being derived or developed. We discuss the cytosolic, in which the splitting of the mesodermal cell takes place, and the enterosolic, in which the pouch like segment of the archenteron takes place and then they split finally to form the Cillum. Next we discuss the segmentation, and lastly we discuss the notochord. Okay, so that's all for today. We will meet in our next class. Thank you.